Hello and welcome to Hydescience Lab Series number 13. This is the Staining Eukaryotic Cells Lab. This is not necessarily part of our biotech standards, but I'd like to do this to kind of prepare us for what is coming that is part of our standards. So our big standard is involving bacteria, and plasmids is a big focus of us for this year. So for our class, gram staining and identification of bacteria is huge. But we do this to kind of get some experience with the staining process. It's not as complicated and gives us some experience with the microscope and focusing in and identifying different features. So it's just a really good lead-up activity to give us some practice before we jump into gram staining and trying to identify different types of bacteria. So without further ado, let's get into it. Alright, so here we go. We have our slide. Not really sure the best way to show this. I'm going to kind of just demonstrate one setup. What we're doing is, this will be a big copy for the students. You see there's 9 different boxes here for our data. So we would be running this trial with onions, cheek cells, and celery. And then we will be staining them with 3 different stains: iodine, fast blast, and Janus green. And we're using 3 different stains because stains have different properties and are used to identify different features of cells. So we have our fast blast stain, which is designed to bind DNA. This is going to show us our nucleus really well and DNA. Iodine stains starch, so it'll kind of highlight different parts depending on the cell. And then Janus green is an excellent stain for staining the mitochondria and allowing those to be visible. We will then sketch as detailed as possible what we see and compare and try to come up with those on our own. So we'll run 3 slides. We're going to divide our slides up into 3 sections. So we'll use a Sharpie to kind of make 3 circles on our slide. We use a Sharpie to kind of, so we don't forget where we put it, so we might not be able to see it, just to kind of help us stay focused. So we'll just say this is the cheek cell. So we'll take a toothpick and we will kind of swab the inside of our cheek, and then we will rub the cheek cells onto the side, get a new toothpick, same process, get a new toothpick, same process. Then we'll come back and we'll drop some different stains on it. So we'll go with the iodine stain, and we just want to put 1 drop on there. We can come back with the Janus green stain, another drop as well. Well, this one might be about empty. Here we go. This one is not empty. So we'll drop a Janus green on there. And then finally we will drop some fast blast stain on our cells. We're just making a wet mount, so then we'll come over and we will drop our cover slips on. We'll kind of drop those down at a 45-degree angle. So this slide is ready for the microscope, in which we would try to study our cheek cells. We'll make one of these slides for cheek cells, onion cells, and celery cells. And we will be cutting samples from those on our own. So just to kind of give you what would we expect to see. So this is kind of what we would expect from the iodine stain, with our onion and celery cells both being plant cells. They would look very similar, but slightly different. And then our cheek cell, being an animal cell, does look different. And so we kind of see, what does it highlight? Is it highlighting little starch molecules at the cell walls? Then we move on to our fast blast, and you can see that it highlights the nucleus very well, because this is where the DNA is located, and that's what this stain does best. And then on our celery cell, this celery is usually Janus green primarily because this is an excellent way to see the mitochondria. We don't usually get good results with seeing mitochondria in the onion and cheek, but we do with the celery, and that's really why we added the celery into this lab. Yeah, so that's it. That's our staining cells, the introduction. So as we get prepared to move on to gram staining, so I will see y'all in that video.